Thanks for providing this opportunity to present our company and also our product. I'm usually from GBIOLOGICS. We are developing this stable galectin-9, shortly SGA9, for KRAS mutant cancer. I'm going to shortly go over this, briefly introduce this product, S-galectin-9, and the efficacy of this galectin-9 toward the KRAS mutant cancer. Okay, galectin is lectin-binding proteins, and it involves in immune homeostasis. There's a lot of galectins in human body, and mainly there's 3 subgroups, and galectin-9 belongs to tandem lipid subfamily. Tandem lipid families, as you can see in the file, there's 2 different lectin-binding active sites linked together by amino sequence, and we call it linker. And as I mentioned, that we are very much among all these galectin families. We are interested in this galectin-9. As you can see, this galectin-9, natural galectin-9, as I mentioned, is linked by this linker, and this linker, this amino sequence, is very susceptible to protease that is easily broken, that these 2 active lectin binding sites separate, then you lose activity. So this linker part was, by engineering, we removed this, modified this protein by removing this linker part, and these 2 active sites put together, and we call it S-galectin-9, stable galectin-9. And it is about 284 amino acid sequence. And this material is patent registered, and we are in very early stage of this product development. Okay, from the articles you can search through that galectin-9 involves, there's many various activities galectin-9 involved. And you can see there's a lot of potential activities and potential indications. Among these indications, we are currently focusing on KRAS mutant cancer. And so I'm going to introduce about the galectin-9 to KRAS mutant cancer. What kind of activities that mechanisms of this galectin-9 has to, for this KRAS mutant anti-cancer efficacy? We mainly have these 3 activities. First one is cytotoxicity. Also, you can find from the various articles that this galectin-9 has cancer cell apoptosis activities. The second one is cancer cell suppressing cancer cell metastasis. Also, you can find articles about this. In addition to these 2 general anti-cancer activities, we have this KRAS mutant cancer cell apoptosis activities. We are focusing on these activities for this KRAS mutant anti-cancer efficacies. In normal cell, when you administer exogenous administer galectin-9, it internalized by endocytosis, and it is involved in regulation of lysosome, that lysosome smoothly function its own function to degenerate material in body and use as energy. However, in KRAS mutant cancer, when you administer this galectin-9, it internalizes and then it accumulates in lysosome. Because it keeps accumulating in lysosome, it swells, vacuolized, and then causes cell death. So we are focusing on this function of this galectin-9 for this KRAS mutant anti-cancer, to develop as KRAS mutant anti-cancer drug. Okay, this is just to show this the study result published by Netherlands scientists. They done this using galectin-9 to see the KRAS mutant colon cancer activity. You can clearly see from this first article that this galectin-9 cause vacuolization and then reduce cell viability. And they also showed that this galectin-9 activity was by this lectin binding site, because when you add lectin to this galectin-9, the anti-cancer activity was suppressed. So also you can see from this from these studies, they also compared this natural galectin-9 with the short galectin-9, the linker-removed galectin-9, that showed that the linker-removed short version of galectin-9 has more higher activity, anti-cancer activity. Also, from using these normal colon epithelium cells, shows that it doesn't have any effect on normal cell, only effect on KRAS mutant colon cell or non-polarized cell. This is also this Netherlands scientist article to just show you that efficacy of this galectin-9. When this KRAS mutant colon cancer cell was SC injected to a nude mouse model to have this ectopic xenograft mouse model, and then they inject this galectin-9, and it shows that the tumor volume was reduced. And also the mouse survival was increased. And then there's no body change, which means that it's non-toxic. So from this study, it shows that KRAS mutant colon cancer, S-galectin-9 has efficacy, potential efficacy to KRAS mutant colon cancer. For this, after looking at all these studies, we are currently focusing on the KRAS mutant cancers, especially pancreatic cancer, because KRAS mutant pancreatic cancer, because pancreatic cancer patients, there's more than 90% of these cancer patients was caused by KRAS mutant in cancer cell. So we are mostly focusing on this pancreatic cancer, and this studies were done in Korea. And you can see that when this MIA-kappa-B, which is the KRAS mutant pancreatic cancer from this KRAS mutant cancer patient, then we injected to nude mouse to develop this xenograft mouse model, and we injected either our galectin-9 in low dose 0.5 milligram per kg or 5 milligram per kg, and then also there's a gemcitabine we used as a comparable drugs. Gemcitabine is currently the standard drug for pancreatic cancer patients. So we use gemcitabine 50 milligram per kg. And then another group, we also have the combination therapy, gemcitabine plus our product, galectin-9 5 milligram per kg. And the result shows that the size of tumor was reduced, mostly therapeutically, greatly reduced by as galectin-9 5 milligram per kg. And we can clearly see, even though the error bar is high, we can clearly see that with 5 milligram per kg, we reached this therapeutic index. And gemcitabine alone doesn't have much effect. But however, when you inject gemcitabine to escaline combination therapy, there's reduction was increased. Tumor cell reduction was increased. Also, in survivor, the escaline 5 milligram kg or combination of gemcitabine with escaline, the survival was the highest. And then the monotherapy didn't much work. Gemcitabine monotherapy didn't work much. So from these studies, these preliminary studies, we can come to conclusion that our S-galectin-9 has a potential for KRAS mutant pancreatic cancer. As I mentioned before, we are in very early stage. The problem with this protein was that the protein was unstable. There's a low expression in our engineering. So we work hard to improve this production, and we are currently have a successful result. And we are in the production development, and we are going to scale up to 200-liter scale up, and use this for the non-clinical and clinical studies. And as I mentioned, we are currently in vitro and in vivo studies with the university partners or CROs. Also, we have currently made a contract with the company for the partnership. And up to previous studies, we are more focused on ectopic xenograft mouse model, which means that we injected cancer cell to cells as C. But from now on, we are more focusing on orthotopic mouse model, much more close to this clinical the cancer model. So we are going to study in vivo, in vitro studies. And as I mentioned, we confirmed efficacy using this the pancreatic cancer cell and colon cancer cell. Also, we filed use as a patent. Our goal is, as we plan to submit IND to US FDA in early 2022. Okay, up to now is about the production. And briefly, I'm going to go over the company, introduce our company. Our CEO is David Song. He has various experience in development of biopharmaceuticals and more than 30 years development and involvement in development of a lot of various proteins, drugs, as well as vaccine. Also, our team members all has vast experience, more than 20 years experience in development of pharmaceutical product. Our company was established in 2018. However, the product was actually, Escarline was acquired in early 2016. And then the company was spin up to GBIOLOGICS, the company right now we have. So now we are developing it, and we got investment. I think Series A was completed. Our company actually has more experience in consulting because we have vast experience in development of biopharmaceuticals. We provided consulting services as well as producing our own product. And then we have vast network as well as partners. And there's the list of products that we involved for the development, from the proteins to vaccines and chemicals. And currently we have in pipeline, we have 3 products. This anti-cancer drug, as well as RA for Escarline product, and then Lucentis biosimilar. Thanks very much.
Okay, thank you for presentation. Okay. Any question or comment? Yeah. Okay. So there is one question. Yeah. Warren?
Post-retirement, JDRetirementGuide.com. No visitors. I'm here as an attorney. In the 60s. Listen to the
Well, it's not clear. Maybe Warren, you can send by chatting room, and we can respond to you.
January 27th on NBC. Women Amplified, a Massachusetts Conference for Women podcast hosted by award-winning journalist and author Celeste Sidley. Visit conferencesforwomen.org/podcast.
So, can you show the slide number 11, the KRAS mutant pancreatic cancer?
Yeah.
In that slide, you show that the Escar 5 mg/kg shows better efficacy than combination with the gemcitabine.
Yeah.
Why is that?
That's very, actually, we expected better with combination, but somehow I think there's interference. I don't know, because the gemcitabine Escarline clearly has 2 different mechanisms. But we are thinking maybe reconfirming these studies to see if there's any interference or the cause of this.
Yeah,
However,
you
it's
should-
better than gemcitabine alone.
You should verify this.
Yeah,
This is very
yeah.
unusual.
Yeah.
Okay, thank you.
Thank you.
Mhm.